In the following, we will focus on 3 conditions: common diarrhea, antibiotic-associated diarrhea, and the effects of antibiotics on gut microbiota. So let us start with common diarrhea. Common diarrhea usually doesn't require an analysis of the patient's gut microbiota, as this would not have any specific impact upon therapy. However, there are cases when stool culture analysis might be needed in order to identify a pathogen or to trace its origin. This is the case, firstly, when diarrhea persists for more than 4 days and is combined with high-grade fever. Secondly, when dysentery has been diagnosed. And thirdly, when an epidemiological surveillance is underway. In any case, diarrhea changes the gut microbiota transiently. While the mucosal content, which is harbored by the outer layer of the mucus, remains mainly unaffected, part of the luminal content is washed out by the watery flux. The slide shows the microbial composition of the 2 layers according to the current state of research. At present, only few human data are available, as the sampling of mucosal content poses considerable challenges. There is evidence, though, that the luminal and the mucosal composition differ from each other, at least at the family and species level. The luminal composition is more variable than the mucosal one, due to kinetics and the transit of ingested, diet-dependent microbes, including probiotics. The mucosal part is harbored by the outer layer of the mucus and is supposed to contribute to the resilience and stability of the gut microbiota. By washing out part of the luminal content of the gut microbiota, local biotopes are displaced downstream, that is, to the lower parts of the intestine. This changes the luminal pH, thus making conditions in the lower gut more acidic. A closer look at the digestive tract shows that the composition of the gut microbiota changes along the way from stomach to colon while its density grows. In the course of diarrhea, these different biotopes forming the gut microbiota can be diluted and washed down from the small intestine to the colon. This might result in the already mentioned acidification of the colon. The gut microbiota is a resilient system. Within a week or so after the end of diarrhea, the previous microbial equilibrium is being restored. The slide shows the results of DNA analysis of fecal samples from 46 children at 3 points in time: during an episode of acute diarrhea, immediately after recovery, and again 3 months later. The columns represent the quantitative development of 3 groups of bacteria. The first one is the Bacteroides prevotella group, whose members produce different kinds of vitamins. The second one is Eubacterium rectale, representing Firmicutes. The third one contains Bifidobacterium and Bifidobacterium longum. These bacteria are used as probiotics, as they can have a positive effect on gastrointestinal health. Hence, they meet the definition of the term probiotic. Recent studies demonstrate that osmotic diarrhea, too, can induce changes in the gut microbiota composition by reducing microbial richness in the lumen and on the mucosa. As this disease is not caused by pathogenic infection, the reduction of microbial variety is solely due to the process of washing out parts of the microbiota. The red and the green curve show the results of measurements of gut microbiota diversity based on a large number of stool samples. As can clearly be seen, osmotic diarrhea leads to decreased phylotype richness of the luminal microbiota. The reduction of diversity caused by osmotic diarrhea is not restricted to the luminal parts of the gut microbiota. It also affects the mucosal microbiota. The right part of the slide shows a small increase of Firmicutes and a number of other species, while on the left side what you see is a large decrease of subgroups of bacteroids. As already mentioned, it usually takes about a week or so to restore the major part of the previous equilibrium of the gut microbiota composition. Probiotics might speed up the process. According to a recent Cochrane review, most probiotics reduced acute infectious diarrhea duration by an average of 25 hours. To assess this time span properly, one has to take into account the large variability of length of diarrhea in control groups, with a duration from 39 to 173 hours. Here we have two randomized controlled trials, serving as an example of how probiotics can shorten the duration of diarrhea. In the first study with 113 children, E. coli Nissle was applied, while in the second one, with 150 children, the probiotic was Lactobacillus casei. The second trial compared a group taking probiotics with two control groups and additionally divided each of these three groups into hospitalized children and those who were monitored in a community health center. The duration of disease in those children who had taken the probiotics was shortest in both subgroups. Some probiotics are recommended for the management of diarrhea by a number of expert panels. These probiotics can be added to the common treatment of the disease, which consists mainly of rehydration. Probiotics offer a safe way to provide the patient with digestible food and to speed up the recovery process. It is assumed that this is mainly due to a quicker restoration of the gut microbiota. A rising concern is the increase in the prevalence of post-antibiotic diarrhea, which is due to the generally increasing application of antibiotics. This development is illustrated by the fact that meanwhile around 15% of all hospitalized patients receiving antibiotics develop some kind of diarrhea. Antibiotics have an impact on the gut microbiota as they're active against some groups of microbes. This applies to most of the antibiotics with a broad spectrum coverage, in particular cephalosporins, fluoroquinolones, extended coverage penicillins, and clindamycin. One of the major consequences of many antibiotics is a transient reduction of the gut microbiota and a weakening of the barrier effect that opens the door for some opportunistic pathogens, for example Clostridium difficile, to proliferate. These changes in the gut microbial composition often lead to disease, in particular to antibiotic-associated diarrhea. Among antibiotics, those with a broad-spectrum antibiotics coverage are the most likely to trigger these processes. Antibiotic treatment decreases the concentration of susceptible microbes, in green, leaving room for the growth of resistant ones, and those that have been repressed so far, in dark blue. The resilience of the gut microbiota aims at restoring a profile that resembles the previous one, at least after a first course of antibiotics. The period of time needed for restoration is likely to be a week or so. But the exact time frame has not yet been established. The more antibiotic courses, the less complete the resilience. The proper treatment of antibiotic-associated diarrhea is not another course of antibiotics, as this would worsen the imbalance of the microbiota composition. Instead, the barrier effect should be restored through intake of probiotics and prebiotics, which are indigestible compounds fermented by some specific subgroups of gut microbiota and thus provide health benefits. Probiotics provide a treatment option that's worth considering. A recent Cochrane review on the prevention of Clostridium difficile-associated diarrhea in adults and children reported that certain probiotics reduced the risk of antibiotic-associated diarrhea by an average of 64%, as indicated by the red rhomb. These are the results of two randomized controlled trials which provide evidence for the efficacy of some probiotics in reducing the frequency of antibiotic-associated diarrhea and of Clostridium difficile-associated diarrhea. For the first one of these trials, with 164 subjects, the probiotic Lactobacillus casei shirota was used, while the probiotic Lactobacillus casei DN114001 was employed for the second one with 135 subjects. With regard to resistant forms of diarrhea, the use of certain probiotics can be beneficial. Let us recapitulate what makes a microorganism a probiotic. It is an umbrella term for living microbes that, when ingested in adequate amounts, can provide a benefit for the host. Particularly with regard to antibiotic-associated diarrhea, or diarrheas caused by C. difficile or other opportunistic microbes, some probiotics are able to prevent the risk and to reduce the incidence of this condition. This is probably due to the capability of probiotics to help restore the microbial barrier of the gut microbiota. Antibiotics have two iatrogenic effects: an imbalance of the gut microbiota and the emergence of antibiotic resistance and its spreading among pathogens and gut microbiota. Increased use of antibiotics causes an increased risk of antibiotic resistance. Let's have another look at this slide that you've already seen. Antibiotic treatment decreases the concentration of susceptible microbes, in green, leaving room for the growth of resistant ones and those that have been repressed so far, in dark blue. This is the first of the two mentioned iatrogenic side effects, the imbalance of the gut microbiota. This slide shows that antibiotics exert different effects on the various components of the gut microbiota. What we can see here is an antibiotic-induced imbalance, called dysbiosis of the gut microbiota. This dysbiosis is associated with the establishment of the bacterial species Enterococci, a total newcomer within the microbial community of these gut microbiota, whose way has been paved by antibiotics. This slide shows long-term impacts of different kinds of antibiotic exposure on the intestinal microbiota. Some antibiotics are active against aerobic, others against anaerobic microbes. As you can see in the middle section of the diagram, vancomycin, for example, decreases some species but not others within the same group. The left-hand side of the slide shows fecal samples of patients who were treated with antibiotics against Helicobacter pylori, while on the right-hand side the stool samples of controls are depicted. By using rRNA sequencing, 4 different phyla were found in these samples: Firmicutes in dark blue, Proteobacteria in green, Actinobacteria in red, and Bacteroidetes in purple. The comparison shows the long-term effect of antibiotics. Over a time span of no less than 4 years, the gut microbiota of the patients lacked stability as compared to the controls. The alteration of the gut microbiota composition has several consequences that are mutually combined. A decreasing concentration of susceptible bacteria and the suppression of their microbial functions, which again influences other components of the microbiota. This concerns, for example, the metabolites of bacteria, which are often feed for other microbes. If these metabolite-producing species are reduced, other microorganisms are deprived of their feed source, which again affects a whole chain of following microbial interactions. Let us turn to the second iatrogenic change that may be induced by antibiotic use: the emergence and spreading of new antibiotic resistance. By metagenomic analysis, the frequency of antibiotic resistance genes within microbes can be monitored. Recent studies tell us that on average antibiotic resistance emerges within 8 years after the use of a given antibiotic. In a sample of 275 subjects from 8 different countries, 53 antibiotic resistance genes to a large number of antibiotics have been detected in the gut microbiota. The most frequently applied antibiotic to which resistance has been developed has turned out to be tetracycline, By now we have reached the end of the second part. You can move on to the next part entitled IBS, IBD, Digestive Symptoms, or to the last part entitled Questions that Patients Might Ask.